Okay, good morning or good afternoon, everyone, and welcome. My name is Giuseppe Iovino. I am a field application specialist at Yikon Genomics, and I will be your host for today's session. On behalf of Yikon Genomics and our partners at NGI, it's a real pleasure to welcome you to Advances in Preimplantation, Genetic Diagnosis, Reproductive Solutions powered by NGS. Whether you're joining from a fertility clinic, a genetics laboratory, or a research setting, we're glad you're here. We have a genuinely exciting session ahead, and we hope you leave with both new insights and practical takeaways that you can bring back to your work. Today, we have two outstanding speakers joining us. First, we have Dr. Antonio Urbano Carrillo, Technical Director at Nuuma Genetics and Professor at Faculty of Medicine at Miguel Hernández University of Elche in Spain. And we're also joined by Elvis Chia, Field Application Specialist at MGI based in Cameroon. I'll be starting today's sessions and will then give a fuller introduction to each of them before their respective presentations, so stay tuned for that. If any question comes to mind as you're listening, please drop them into the chat at any time. We'll be collecting them throughout and we'll have a dedicated Q&A session at the end where our speaker will address as many as possible. So I will be starting with my presentation. And with today's talk, I will present some of the main reproductive solution that Econ offers to improve IVF outcomes. I am currently based in Cambridge, UK, and hold a master's degree in molecular biology and genetics. Before joining Econ, I developed expertise in library preparation and next generation sequencing, first as molecular biologist and then as technical application scientist for more than 5 years, supporting global customers across different workflows and sequencing platforms. I currently help customers within Middle East and Africa succeed thanks to Yikon Genomics. Today's presentation includes an overview of Econ Genomics, and then I will discuss about the core technology behind our solutions and the range of products that we offer, presenting some exciting innovations. So, Econ Genomics is a high-tech enterprise dedicated to the development and application of single-cell whole genome amplification and sequencing technologies, and provides precision medicine solutions in the field of reproductive genetics. Our key solutions include genome testing for parents, embryo, and endometrium testing that either individually or combined, they ensure the best outcome for couples in need. Econ is present in over 44 countries with headquarters in Suzhou, China, and laboratories and warehouses in Spain, Thailand, Malaysia, Japan, and USA. In our labs, we're able to receive customer samples to provide an end-to-end service to those that prefer outsourcing their tests. But the network expands to many more countries thanks to our trusted distributors covering all continents. To date, Econ has helped more than 680,000 patients, and these numbers keep growing. Econ was founded in June 2012 as a result of research studies done at Harvard University, and in December of the same year, the technology was applied to map a single sperm gene with wide coverage. In 2013, Econ started commercializing its first whole genome amplification kit, and in 2014, the world's first baby screened using Econ technology was born. Since then, the company kept expanding its size and portfolio. In 2019, the ERT product was launched, constructing the comprehensive genome, embryo, and endometrium product ecosystem. And just last year, a new pioneering solution for the detection of chromosomal structural abnormality hit the market. As of today, Econ owns a large number of patents and copyrights. It's featured in hundreds of scientific papers and thousands of citations. And last but not least, with CAP accredited labs, GMP certified factories, and ISO accredited management ensures reliable, high-quality services globally. Let's move now to our core technology called Malbak. Malbak stands for multiple annealing and looping-based amplification cycles and utilizes a mixture of DNA polymerases with strong strand displacement activity to carry out quasi-linear pre-amplification cycles of the genome. Additionally, Malbak primers are designed to incorporate specific sequences at the end of the DNA amplicons that enable the formation of loops. The amplification is initiated with a pool of random primers, each having a common sequence and 8 variable nucleotides that can evenly hybridize to the genomic DNA. The DNA polymerase with strand displacement activity are used to generate semi-amplicons with variable length, which are then melted off from the template. Subsequent amplification of the semi-amplicons then gives us full amplicons, which have complementary ends that allow the looping, preventing further amplification and cross-hybridization. The next amplification cycles are then repeated using exclusively the original genomic DNA and the semi-amplicons, eliminating the sequence-dependent amplification bias typical of the PCR-based methods with random primers. With a quasi-linear amplification, if there is an error in one copy only, it becomes negligible in the amplification product. The resulting loops are then further amplified for next generation sequencing. And the result of Malbak is therefore a low allele dropout and a high genome coverage. As shown on the left of this comparative table, Malbak technology has proven to give better uniformity of genomic amplification compared to other solutions in the market. On the right-hand side, we can see how Malbak performs better in terms of allele dropout rate and reproducibility. Starting from this successful technology, Yikon has developed a large range of products that cover the most common and modern applications in reproductive medicine. Starting with three of the main solutions that test embryo and parental blood in some cases, being PGTA and PGT-SR for the detection of all chromosomal abnormalities with a single workflow, PGT-M and its innovative SNP-based solution to overcome current barriers, and Kairos-seq, an integrated solution for PGT-M, PGT-SR, and PGTA. Our PGTA and PGT-SR test rely on chroMIST, a product that combines genome amplification and library preparation in one step, reducing the preparation time to 2.5 hours. In this way, the entire workflow from cell lysis to data analysis can be completed in 11 hours. And in this regard, I will mention now our ChromeGo data analysis platform that allows to process samples within minutes and provides pipelines for every Yikon solution. I will talk about ChromeGo in more details later, but for now, I want to highlight the flexibility of chroMIST that is adaptable to different sample throughputs and resolution requirements, and its reliability being validated with more than 100,000 embryos. For PGTA, Yikon offers three solutions that differ in the information provided, the amount of data, and the sample type. These are PGTA Basic, Upgrade, and Complete. They all make use of the same library prep kit, our chroMIST. And depending on the number of reads per sample, we have PGTA Basic that requires between 200,000 and 1 million reads, corresponding to chromosome level and 4 megabase level deletion and duplication resolution. The sequencing requirement is just 55 base pair single end, and the detected content is aneuploidy at these different levels, as said before, gender, mosaicism, and sibling QC. Sibling QC is a control step that checks if two or more embryos are related or not. Next, we have PGTA upgrade with 3 million reads per sample. Same sequencing strategy, but additional content such as triploidy, uniparental disomy, and more than 42 CNV-related syndromes. The last solution is PGTA complete that requires 20 to 30 million reads per sample, parental blood together with the embryo cells, and provides additional information on the origin of aneuploidy, kinship analysis, and origin of mosaicism. I would like to add that as part of the flexibility feature, all Yikon's products can be multiplexed in the same run. All our products are compatible with all MGI instruments indeed, including the E25 platform. We have recently launched our Chromis protocol for E25, and the data produced by our team show that this instrument performs better than some of its siblings, like T7. All karyotypes of the control samples tested were indeed confirmed on E25, demonstrating strong efficiency and reliability of Chromis libraries on MGI sequences. You can see here how 3 of the samples tested did not produce the expected results on T7, while they were correctly reported with the E25 sequencing. With the same libraries prepared with Chromist and the same sequencing strategy, we can also detect other types of unbalanced chromosomal rearrangements, such as insertions, down to 1 megabase of resolution. Therefore, customers can have 2 tests in one, analyzing the same data with 2 different pipelines. Regarding the balanced rearrangements, instead, these are detected by the Carrier-seq solution that I will talk about in a minute. Moving on to the detection of monogenic diseases in the embryos, some of the most prevalent challenges come from the little availability of DNA in single cells or embryo biopsies, and the risk of allele dropout due to factors such as insufficient template or diagnostic method efficiency. These can easily be translated into misdiagnosis with subsequent pregnancy failure or defective embryo. As previously discussed, our Malbach technology can, yes, reduce ADO, but cannot eliminate completely. Additionally, different types of mutations, such as point mutations or copy number mutations, require different diagnostic methods, and a single whole genome amplification product cannot accommodate for the different requirements of these methods. So the solution that Yikon developed to overcome these obstacles make use of the SNP linkage analysis, namely the coexistence of multiple SNPs within one chromosome, also known as haplotype, instead of the single SNP of the monogenic disease. Once a mutation is associated to a specific haplotype in the parents, the identification of the pathogenic sites in the embryos is translated into the detection of the entire chromosomes. In this example, we see a case of autosomal recessive allele carrying the pathogenic mutation. If we are able to infer what maternal and paternal chromosomes carry the mutation based on the upstream and downstream SNPs, then we can remove the risk of ADO when testing the embryo because we essentially have an entire set of other SNPs available. To assign a mutation to a specific chromosome, however, we require a complete family pedigree that includes parents and affected child, or parents and grandparents. This is due to the limitation of NGS technologies, where the read length is not sufficient to construct the parents' haplotypes. Therefore, we need to rely on inheritance patterns using DNA from additional family members. The PGTM workflow is then simply divided into 2 parallel tests, one for the pedigree or the family, and one for the embryo. In both cases, a SNP linkage analysis is performed with an additional aneuploidy screening for the embryo, which can be either PGTA basic or PGTA upgrade, depending on the reads number. Our PGTM test requires more than 2 million reads per sample. For those professionals that require multiple PGT tests, Yikon has developed Carrier-seq. This is an all-in-one, meaning that the same library is used for multiple applications, such as PGTA, PGTA-SR, and PGTM. Having such solution allows to greatly reduce operation time and reduces and resources as only one sequencing run is required for all tests, but also the cost per sample. This solution requires both embryo biopsy and family blood and uses Chrome Swift, a kit based on Malbak, just like Chrome Inst, but better suitable for this application. And the main benefits is the flexibility of having multiple tests in one library and one sequencing run. The real magic happens indeed at the bioinformatics level. Firstly, sequencing requirements are a paired end run and 20 to 30 million reads per sample. Of the generated reads, only some are used for the CNV analysis, as these only requires little coverage, while a larger amount of data is used for analysis of heteroploidy origin, kinship, and SNP linkage analysis for PGTM. The turnaround time from sample preparation to report in this case is longer, but doctors are provided with all information at once. Other products offered by Yikon that focus on parental testing are ERT or EMT for the prediction of window of implantation and microbial characterization in the endometrium, carrier screening for the detection of monogenic disease carrier status in parents before pregnancy. And I will also briefly talk about NICS, the world's first non-invasive embryo chromosome assessment using spent culture medium for improved IVF outcome. The solution that Econ offers for endometrium receptivity test is called Essence, and it was developed to tackle two major issues: the detection of window of implantation misalignment and the endometrial dysfunctions. The test therefore provides a clear confidence interval for the best window of implantation by the hour to guide safe transfers. In addition, information on epithelial subtype, composition, and immune status for further treatments or recommendations. The test is aimed to those patients that have experienced repeated implantation failures, patients with precious or few PGT embryos, and patients with high risk of window of implantation displacement due to polycystic ovary syndrome, diminished ovarian reserve, endometriosis, endometriosis, etc. The test makes use of endometrium biopsy or uterine where the RNA is extracted from and converted into cDNA. From this point on, standard WGA and library preparation are performed, followed by sequencing and analysis. The test only needs 1 million reads per sample, which allows the testing of a large number of samples in a single run. The technology behind the data analysis of the window of implantation prediction is based on the artificial intelligence and a model built considering more than 200 differentially expressed genes involved in different processes during embryo development. The expression level of the selected genes classify the endometrium in receptive, post-receptive, pre-receptive, and proliferative, where proliferative means that it is in the non-receptive stage, and it is recommended to contact the physician to confirm the sampling time. Additionally, a similar model was used to analyze more than 100 immune genes and the pro-inflammatory and anti-inflammatory cells. This feature was developed following some findings that demonstrated how an excess of pro-inflammatory cells is associated with low clinical pregnancy rate. In the same way, an abnormal stratification of the epithelium can lead to poor pregnancy outcome. For example, a study showed how lower proportion of unciliated epithelial cells translates into reduced pregnancy rates. Remaining in the topic of endometrium, I would like to introduce our EMT solution that stands for endometrial microbial test. The concept behind this product is that in healthy women, the diversity of microbial composition gradually increases, going from the vagina to the ovaries. The species of Lactobacillus, in particular, play an important role in maintaining a healthy microenvironment in the reproductive tract. It can reproduce lactic acid that maintains the acidic environment of the reproductive tract and inhibits the growth of harmful bacteria. If the lactobacillus content is low, the possible consequences are reduced implantation rate, reduced pregnancy rate, and increased number of abortions. EMT uses 16S ribosomal RNA to determine whether the uterine microbial environment is optimal for endometrial health. Our analysis pipeline provides information about the present species, such as identification and proportion, with focus on Lactobacillus, and if there is any pathogenic bacteria. The third product dedicated to parent testing is our carrier screening tests that are generally offered to individuals or couples with a known family history of a particular condition or from a particular ethnic background that is known to be at a higher risk of inheriting a particular condition. Yikon offers two main configurations, light and expanded, that cover respectively 36 diseases in 31 genes and 566 diseases in 495 genes. Finally, the solution I want to introduce is called NICS-INST and was developed to replace traditional PGTA tests and process embryo culture medium for library preparation instead of embryo biopsy. The library preparation steps and duration are the same, and data show high correlation with standard PGTA methods. Blastocysts generally release a given amount of DNA into culture medium during the development, and Yikon has successfully built a strategy that makes use of this released DNA to test embryos for aneuploidies, gender, mosaicism, etc. You can easily understand the benefits of using a culture medium instead of cutting cells out of the embryo. The risk of damage are greatly reduced while maintaining the same reliability. Here, for example, we can see the results of a study that compared sequencing data obtained from biopsies and the corresponding media for a couple where both partners were affected by abnormalities. The correlation between the sample types, so trophectoderm biopsy and culture medium, was confirmed, and the embryo selected from the analysis of a culture medium resulted in a successful pregnancy and birth. This was the first NICS clinical case for Yikon in 2016. But most recently, Yikon has integrated NICS with an AI embryo grading system that looks at features like chromosomal mosaicism proportion, segmental aneuploidy size, multiple chromosomal abnormalities, and sex chromosome information to classify the embryos in grade A, B, and C, from those with the highest euploidy probability rate to those with the lowest. The algorithm was fed with pre-existing data and outcomes, and a retrospective analysis was performed using the mentioned parameters. The study showed how with traditional non-invasive PGT methods, the embryo's utilization rate was of 57.9%, while with the NICS AI system, the rate increased to 78.8%, therefore with an improvement of 20%. The immediate benefit of this technology is a strong reduction in embryo waste, giving patients more opportunities to successful pregnancies and births. Lastly, a quick overview of what our ChromeGo platform looks like. I want to highlight here the complete customization and flexibility of selecting what resolutions to investigate, what information to report or not, such as sex and mosaicism, and finally the possibility to customize the mosaicism ratio range based on the local requirements. This is an example of what the results view looks like with information about the data QC, the CNV result, sex chromosomes if reported, the karyotype, and graphic visualization of the ploidy for each chromosome. This is to demonstrate the clarity of the information you can find on our platform. And with this, I conclude my presentation. If you have any question, as I said, just drop them in the chat and we'll answer at the end of the session. Thank you. We are going to move on to our next presenter, Elvis Chia. He graduated from KU Leuven University with a master's in clinical molecular biology in 2015. And before joining MGI, he's been active within molecular biology diagnostics and population genomic studies in the UAE. He now supports customers in Africa across different sequencing applications. Elvis, I hand it over to you. Feel free to share your screen.
Okay. Thank you. Good morning, everyone. Please permit me to share my screen quickly, or just share my window. Yeah. So can everyone see my screen, please?
We can see the PowerPoint application. We don't see
The
the full
PowerPoint.
screen.
Okay.
Yeah.
Yes, the PowerPoint. Okay. In
Okay.
presenter mode. All right. Good morning once more, everyone. First of all, thank you very much for sharing this space with us and for the opportunity to present how MGI technology, together with the applications developed by Yikon, can make a difference in reproductive genetics analysis. It's a great pleasure to me to share this stage with our distinguished high-level speakers, as well as with all of you. That said, we would like to begin with a brief introduction of MGI and our parent company, BGI. Oh, sorry. Yeah, sorry. Oh, I was filming the last slide. Sorry. Okay. Um, As many of you probably already know, MGI belongs to the BGI group, the world's leading provider of sequencing and proteomic services. Our main objective is to bring benefits to people by providing academic institutions, pharmaceutical companies, healthcare providers, and other organizations and solutions across a wide range of applications. BGI was founded in 1999 in Beijing within the framework of the Human Genome Project, being one of the main participants by sequencing 1% of the human genome. So what began as an institute solely linked to this project has grown exponentially to the present day, now empowering over 10,000 people, holding more than 1,700 patents with over 3,400 scientific publications and 400 plus scientific papers in top journals such as Nature, Science, and Cell. Additionally, the new headquarters, sorry, excuse me, sorry. Oh, yeah. Hello, can you see my screen? Yeah.
Elvis, we can only see the first slide.
Oh my God. This is strange.
So we're not seeing the presentation mode. We are seeing the entire PowerPoint app with all the slides on the left.
That happening. Um, you can't see subsequent slides. No, it's not playing. You can't see my next slide.
We can see the slides at this moment, they are on the left, but we see all the slides. It's not the presentation mode. So right now you are on slide number
I
7.
don't know why, but my slide is on. It for me. It's on presenter mode. One moment. I do. Sorry, just one moment. Pasha is trying to call me. Yeah, but I'm on slideshow. I don't know why it's doing that actually. Okay, let me try this from the beginning. Hello? Is it better now? Can you see it now?
It's not presentation mode.
It's still not presentation mode.
No.
Why is it? Because I am. Can you see my slide? Can you see my screen?
Yes.
Like with the window. Yeah, this is slide share, right? This is. I'm clicking. I click here, so it should go
You
to
can
the
press
beginning. I
the
don't know why.
current slide.
Okay, can I send it to you? Please, Giuseppe, can I send this PowerPoint to you so that you can present it for me? Because I don't know why I am. It's not going. I don't know why it's not doing the play mode.
Oh, okay. And you can share your file to me and I can share it for you.
Yes.
Yes, please. Just one moment. I
Okay.
can send it to you email right now. Sorry. Oh, I'll just send to Rainy. Oh, blah. Where is it? Okay, you what? This, my God. I'm sending. Oh, so you can get a copy. Maybe I can share on WeChat. That. To this one. Just come here. I'll just share here.
Okay, sorry. Dr. Antonio, could you start your speech first? We can let Elvis be the last one.
Okay, no problem.
Thank you.
I sent it. I shared it. Don't know if you will see it.
Yes, I received, but we can let Dr. Antonio speak
Okay,
first. Yes.
can I start
Okay.
my presentation then,
Thank
no?
you. Yes.
Okay, let's go. Sorry, here. Can you see my screen and my presentation?
Yes.
Okay. Hello, thank you so much to Yikon Genomics to invite me to this session. I want to share with you our experience, and I want to show some tips or some things about carrier screening in assisted reproduction that is a topic that I'm deeply passionate about because it's changing the way we think about reproductive medicine, and this is the way that we can help couples that go towards centers to get some help. To understand why genetic carrier screening matters, let's look at a few facts. Okay, the human genome has around 20,000 genes, and we currently know over 7.1 genetic conditions linking to variants in those genes, a number that keeps growing as research advances. About about 4.5 thousand conditions are caused by a single gene and follow a recessive or X-linked inherited pattern. What makes them particularly challenging is that they can be transmitted by two completely healthy parents with no family history of disease. That's not an exception. That's the rule of these conditions. And that's precisely why preconception screening exists, because 1 to 3% of couples present a high genetic risk for these diseases. These conditions affect about 3 or 5 in 1,000 neurons. And when we look at pediatric hospital impact, it's striking because up to 40% of pediatric emergency causes of genetic origins and up to 71% pediatric hospital admissions are from this kind of genetic conditions. Genetic is not rare, it is everywhere in pediatric medicine. In practical terms, this means that 1 or 2, 3% couples are in high genetic risk, or is the same that 1 in 40 healthy couples with no family history or with another symptoms are at a high genetic risk and have a risk to have a child affected by one of these diseases. This is a silent transmission of this risk. Okay, to put this into the context, when couples think about genetic risk, when couples think about genetic risk, Down syndrome is always their biggest fear, and it is unstable. But the data tells us a different history. The actual risk of having a child affected by a recessive or X-linked genetic disease is higher than the Down syndrome, and these conditions are more common yet far less now. And that is precisely the gap that the genetic carrier screening is designed to close. To understand why carrier screening matters, we first need to understand how these diseases are transmitted. Each of us has two copies of a gene, of every gene, one from mom, one from dad. In autosomal recessive conditions, a child needs to inherit a fault copy from both parents to be affected. The parent each carry just one, so they are healthy, with no symptom and often no family history of the disease at all. But with every pregnancy, the odds are the same. 25% or 1 in 4 chance of an affected child, 50% chance of a healthy carrier, and 25% of completely unaffected son, child. Sorry. Here. X-link conditions follow a different logic, and sex is at the center of it. Males have one X and one Y chromosomes. Females have two X chromosomes. When a pathogenic variant sits on the X chromosomes, males have nowhere to hide. 140X is enough to cause the disease. Females with 2X chromosomes are usually protected, but they carry the variant silently and can pass it on. For a carrier mother, every son has a 50% chance of being affected. Sons have being affected, and every daughter has a 50% chance of becoming a carrier. No family history, no symptom in the mother, and yet with every pregnancy, the risk is there. This is why X-linked conditions are a critical part of any carrier screening panel. The third pattern is autosomal dominant inheritance, and I'll cover it briefly because it sits outside the main focus of today's talk. In dominant condition, one faulty copy is enough. This disease appears in every generation, affects both sex equally, and carries a 50% transmission risk with every pregnancy. Huntington disease and Marfan syndromes are classic examples. These conditions are not the primary target of carrier screening, and for good reason. The variant doesn't hide. The affected parent is symptomatic. The family history speaks for itself, and couples are usually already aware of the risk before they have even considered pregnancy. Carrier screening exists precisely for the opposite scenario, when the risk is completely invisible. But here's the good news. We now have the technology to change this. Most of these conditions are severe, disabling, and without effective treatment. We cannot cure them, but we can prevent them. And prevention starts before conception. Genetic carrier screening is how we do it. A simple test, one sample, one result, that can identify whether a couple is at risk of having an affected child. Now after the diagnosis, not during the pregnancy, before. That shift from reaction to prevention is what makes carrier screening one of the most powerful tools in the modern reproductive medicine. Now that we understand that we are looking for, let's talk about the tools we use to find it. The first is the most traditional, clinical and family history, only a paper and a pencil, a 3-generation pedigree assessed case by case. This is very important. This is a simple, low touch, and still valuable information that we have to take from the couples, especially for identifying dominant inheriting patterns where the family history speaks for itself. The second tool is cytogenetics. In cytogenetics, we can do a karyotyping and fish on sperm. These techniques allow us to detect chromosome anomalies and structural rearrangement that molecular methods cannot see. They are not replaceable today, at this moment. And third, the gold standard today is molecular genetics. Next generation sequences, or NGS, allow us to analyze hundreds of genes simultaneously at high resolutions. Combined with MLPA for copy number variation and TP-PCR for conditions like Fragile X, we have a comprehensive toolkit that covers the vast majority of clinical relevant genetic risk. The key message here is that no single tool does everything. A complete evaluation uses all 3 levels, from the patient family tree to the sequencer. And there is one of the key questions that I show you today. How many diseases should we screen for and which ones? This is one of the most important questions in our field. And I'll be honest, there is no single correct answer. Technically, we could sequence the entire genome, but that could not be responsible medicine. The goal is balance, genetic security, clinical utility, and cost, of course. We rely on international guidelines and on the experience of groups who perform this testing routinely. So, how we decide what disease to include? International society agree on 3 main criteria. First, the condition must be severe. Sorry. We are talking about chronic progressive disease, conditions that affect a child's ability to move, think, or perceive the world, not mere conditions, not adult onset conditions, severe early onset disease with a clearly established clinical picture. Second, the variant must be confirmed. We only report pathogenic or likely pathogenic variants, classified according to established international guidelines, not variant of uncertain significance or boost. Certainty is what allows couple to make decisions. And third, the conditions must be pan-ethnic. It should be relevant regardless of where the patient comes from. Carrier screenings today must move away from ethnicity-based testing because in a diverse world, mobile world, ethnicity is no longer a reliable predictor of genetic risk. These criteria are not arbitrary. They are designed to give couples actionable information that can you actually use. Now let's look at the other side, what we should not include. And this is just as important as know what to include. We exclude conditions where the evidence is limited, very few published cases, little data on survival, variable expression, or incomplete variable expression, that meaning that the same variant can cause very different symptoms in different people. Incomplete penetrance, meaning that some carriers never develop the disease at all. That kind of uncertainty is not useful for reproductive medicine and decision making. We also exclude conditions where the genotype-phenotype correlation is poor. If we cannot predict what a positive result actually means for that child, we should not report it. We exclude low penetrance risk variants. These are variants that increase the relative risk of a disease, but do not guarantee it. This is not the same as a recessive condition. It is a different clinical scenario entirely. We exclude dominant conditions, we exclude mitochondrial disease and polygenic risk disease, and we exclude benign condition, of course, variant of uncertain significance. The mission here is simple. We only report that we can act on. Information that creates anxiety without enabling decisions is not a good practice. Now, let's talk about the specific recommendation from international society, because this is where the science becomes clinical practice. The first recommendation is about terminology. The term expanded carrier screening is being replaced by simple by carrier screening. Why? Because the word expanded implies something optional or extra. It is not. It should be a standard. Second, it established a tiered system. Not all patients need the same panel. We organize conditions by prevalence and inherited pattern, and we offer the appropriate tier based on the clinical context. Three, it must be ethnicity neutral screening. This is fundamental shift. We no longer ask where a patient comes from before deciding what to test. Pathogenic variants do not respect ethnicity boundaries, and neither should our screening partners. Fourth, and this is the key recommendation, Tier 3 screening should be offered to all patients with reproductive intent, not only for reproductive assist patients. That is, be offered to all patients with reproductive intent and their male partners. Tier 3 means condition with a carrier frequency of at least 1 in 200 plus X-linked conditions. And finally, panels covering condition rare that 1 in 200 should only be recommended in specific cases, consanguineous couples or patients with a relevant family history. The bottom line, carrier screening should be universal, ethnically neutral, and offered before conception. These recommendations come from this paper published in 2022, and I encourage you to read it. It is one of the most comprehensive updates on carrier screening available, and it forms the scientific basis of everything we discussed in this last year. Let's look at what the market offers today. As you can see here, commercial panels vary considerably in size, from around 300 to 550. All of them report pathogenic and likely pathogenic variants only, and include CMV analysis for key genes like SMN1, and HBA for alpha thalassemia, and all includes special studies to foreign conditions like Fragile X. But there is the most important number of this slide. This bar chart on the right and here, across all commercial panels available, only 89 genes appear in the same gene, in every single one. The total number of different genes across all the panel combining is 830, and only those 89 genes are in all the panel. What does that tell us? Those 89 genes are the scientific consensus, the conditions that every laboratory, every guideline, and every clinical groups agree must be included. Everything beyond that 89 genes is a decision, and the different groups make different decisions. And Nuuma Genetics and Yikon, like Giuseppe told us before, we have two different solutions, two different carrier screening, while light with about 36, 37 genes, and one more comprehensive, going about 560 genes. This more means better. This is one of the most debated questions in our field, and the data give us an announced answer. Look at these three that studied in this table. Martin et al. Analysis 549 genes in 2,500 patients and found that 84% of them carry at least one pathogenic variant. Abuli and her group used 200 genes in 1,300 patients, and she found 56% of carrier. In our group, we detect about 20% carriers when we study a panel of 15 most important genes. And more genes means more detection, but it also means more variants of uncertain significance and more complexity in interpretation and more need for strictly genetic counseling before and after the test. More is not always better. Here is the example of the genes that must be included in these 89 genes that are in all of the commercial panel that they offer right now. Okay, that Giuseppe told show us before. Nuuma Genetics and Yikon have these two panels, one with 566 disease, about 500 genes, and separated in 16 disease systems. The little one is this is an update. Sorry, the Giuseppe is the that right now we are using, and is about 36 disease. So when is the right time to perform carrier screening? The answer is clear: before starting any reproductive cycle. Preconception, not during pregnancy, is before. Why? Because if we identify a high-risk couple during the pregnancy, the options are limited and the decisions are much harder. Preconception and testing gives couple time to think, time to plan, and time, the most important time, to choose. Now, how do we do it? There are two strategies. One is we can do it simultaneously. Both partners are tested at the same time. This saves time. If you are working against a tight reproductive calendar, this is preferred approach because you know that the time is gold when we are talking about reproductive assisted technology. The second strategy is sequential testing. We start with the woman, always with the woman first, because this allows us to cover the X-linked conditions. If her result is negative, the couple is considered low risk and we can proceed. If her result is positive, we then test the male's partner for the same gene or the same panel. Sequential testing can reduce cost because if the woman is negative, there is no need to test the male partner at all. In practice, the choice between simultaneous and sequentials depend on time and cost, but the time in preconception is not negotiable. Here we have a Beneruso work that show in a diagram the most important thing that we are treating is in this scenario. Couples that planning a pregnancy, couples who will carry an assisted reproduction, and couples who choose to have preconceptional screening or consanguine planning a pregnancy. This is the more common scenario that we have to do or to have to make a carrier screening, and when in preconceptional period, okay? And you have to remember that the recommended is tier 3 unless you are consanguinity planning pregnancy, that is better than a more extensive test. Here we have the scan of possible outcomes when we can encounter in a sequential study. We start with the woman. If the women have no pathogenic variation, we can continue the cycle with the security that they have a low risk couple. But with pathogenic variants, we have to study the partner, the male partner. If there is no mutation in the same gene, this is a low risk couple. We continue the cycle. If we have the mutation in the same gene, this is a high genetic risk couple, and we have two ways. One, for pre-GTN, we can use Cariosec, for example, or maybe we can choose a gameted donor. But it's really important that the gameted donor have the same carrier screening that we use here. And I want to introduce you in this slide a concept that is becoming increasingly central to reproductive medicine, that is the genetic matching. Genetic matching means ensuring that a couple or a patient and their donor do not share pathogenic variants in the same genes. It is not just a screening, it is compatibility testing at the genetic level. The concept is particularly powerful in the context of gameted donation, because it is where we have the greatest ability to act. We can choose a donor, we can change a donor, and we cannot change a patient's genetics, but we can find the right one for them. And when we get the match right, we offer in the couple something extraordinary, the greatest genetic security that medicine can currently provide. Not zero risk. Really, there is not zero risk, but as close as we can get. I want to show you a different scenario for gameted donation. There is very important, the carrier screening gameted donating program, and there is the first one. This is the most common one. If both the donor and the patient undergo a carrier screening with the same panel, okay, the results show that the donor carries a pathogenic variant in the gene A, but the patient doesn't carrier a variant in that same gene. The genetic matching result compatible, and these two we call it a low risk couple, okay, and they have a very low risk, a residual risk that we comment in the next in a few slides. In the second scenario, and this is where the genetic matching truly earns its value, both the donor and the patients are tested. This time, the result is different. Both carry a pathogenic variant in the same gene. They are incompatible. Together, they could have a 25% chance of having an affected child with every pregnancy. That is not as stable when we have the ability to act. The decision is clear. Change the donor. Find a compatible donor. This is not a setback. It is the system working exactly as it should. The patient reproductive journey continues safely with a donor who does not share the genetic risk. This is the power of genetic matching in gameted donor. And the last scenario and the third is the straightforward but important, that both the female donor and the patient undergo carrier screening. The results show that the donor carries a pathogenic variant in an X-linked gene. The patient is negative, it's wild type. In these cases, we don't need to compare the two results against each other, as we would in an autosomal recessive scenario. The decision is mediated. The donor is discarded. Why? Because a female donor carrying an X-linked variant cannot donate it safely. Her male offspring will have a 50% chance of being affected, regardless of the patient carrier status. The risk is in the donor alone. This is a clear, non-negotiable clinical decision. Discard the donor, find a compatible one, and the patient reproductive journey continues safely. Okay? And I want to talk something very important, and I think that is very important that the clinician, the physicians, have to transmit to their patients. This is the concept of the residual risk. The residual risk is the risk that remains after screening, even when the result is negative. And it is for basically or mainly for 6 reasons that you can see here in the slide. First, not all of the causal variants are known. Science is still discovering new pathogenic variants every year. The second one is the other unstudied genetic causes may exist. Okay, we can't study all the possible causes of the disease. Third, there is the possibility that germline monocytes, a variant present only in some cells. When this is in the ovary of the testis, this is the possibility that we can detect, and it have a risk of transmissions. Fourth is the novel mutations, new variants that appear for the first time in the child, not inherited from either parent. No test can predict this. No preconceptional test can predict this. Fifth, not all pathogenic variants are covered by every panel. And sixth, there is technology limitations, high sequencing homology, pseudogenes, homopolymers, regions of the genome that are difficult to sequence and reliable. The key principle is this: no genetic test eliminates risk entirely. What screening does is dramatically reduces. That is what we promise our patient, not zero risk, significantly reduced risk. There is a difference. And being honest about that difference is what will be with the trust. Okay? And this is, and I am finishing, and this is this is where everything we have discussed today comes together. Screen, match, and plan. Test both patient and donor with the same panel. Identify compatible donors free of shared pathogenic variants. We can make a PGTM if there is a patient with the same mutation or the same gene mutated, sorry. And this is the plan that we have to trace with this couple. And couples come to us with hope, that I said at the start of my speech, my talk. Our job is to make sure that the hope is built on the strongest scientific foundation that we able. Carrier screening is how we do that. And finally, to key takeaway, let me close with the numbers that matter most. Between 1 and 3% of couples share a pathogenic variant in the same gene. That is 1 in 40 healthy couples with no symptoms, no family history, and no warning. But when we identify those couples before conception, the impact is dramatic. Studies show that the reduction of between 50 and 90% in the incidence of serious conditions like cystic fibrosis and beta thalassemia, and perhaps the most powerful data point of all comes from the Mackenzie Mission Project in Australia. More than 75% of newly identified carrier couples took action to avoid the birth of an affected child, not because they were too tall, because they have the information and they choose to use it. Then it's what carrier screening does. It does not take decision away from couples. It gives them the knowledge to make these own decisions. And the 3 final points to take home. The first is that carrier screening is not diagnosis. It is identifies carrier status. It doesn't tell us that a child will be affected. Not all genetic conditions are covered by a single panel. Limitations exist, and we must be transparent about them. Second, a positive result is not the end of the road. If a recessive mutation is found, genetic matching with a compatible donor can eliminate the shared risk entirely. For X-linked mutation, PGT-AIM or donor chain is the recommended path. There are always options. And third, in Nuuma Genetics, all donors are screening with the same panel as a patient. That means our results are compatible with virtually every panel of the market. No gaps, no surprises. Carrier screening works. Carrier screening works. The clinical impact is real, and the technology is available today. That's all I want to show you, and I'm happy to take any question. You can reach me at my email is here, urbano@nuumagenetics.com, and it's my pleasure. Thank you.
Thank you, Dr. Antonio.
So, can I start?
Yes, we can see the presentation mode now.
Okay. So, you're gonna help me to swap, right?
Yes.
Okay. Thank you. So again, good morning again. I want to appreciate everyone for this opportunity and for sharing the stage with Yikon. Oh, just go straight to the next slide. Please, can you go to the next slide? Yeah, as many of you probably already know, BGI belongs to the BGI group. MGI, sorry, belongs to the BGI group, which is the world's leading provider of sequencing and proteomics services. Our main objective is to bring benefits to people by providing academic institutions, pharmaceutical companies, healthcare providers, and other organizations with integrated genomic sequencing. Looks like there's an echo. I don't know if you can hear an echo. I can hear an echo. Is the sound okay with you?
There is an echo. Are you connected from two devices or just one?
Wait, just one moment. I will just. Yeah, actually, I was connected to two devices. Let me just turn off the other device. Okay. It's better now, right?
Yes.
Yeah. So BGI was founded in 1999 in Beijing within the framework of the Human Genome Project, being one of the main participants by sequencing 1% of the human genome. So what began as an institute solely linked to this project has grown exponentially to the present day, now employing over 10,000 people worldwide, holding more than 1,700 patents, and with over 3,400 scientific publications and over 400 scientific papers in top journals such as Nature, Science, Cell. Additionally, the new headquarters recently opened in Shenzhen is specifically focused on MGI. Please, can you go to the next slide? Thank you. So within this growth, there are 9 entities within the BGI group, spanning areas such as research, education, biobanking, agriculture, and more, covering all aspects of health and research requiring sequencing services. Within this structure, which may seem complex, there are 2 fundamental pillars on which the group is based. 1, We have the BGI Research, which is the largest private research center in China, where basic research is carried out that is later applied to the technologies and tools of the group's different entities. And the second, which is the MGI itself, which is the company responsible for providing the technological tools needed across the group. Please, next slide. Thank you. Focusing specifically on MGI, we are dedicated to the development, production, and implementation of sequencing technology. We are a team of close to 2,700 people, 32% of whom work in R&D, reinforcing the company's strategy of investing heavily in innovation and development. We also hold about 1,000 active patents, and we serve over 3,300 global users. And we have installed more than 4,500 sequencing platforms. And of course, have over 10,900 papers published using our sequencing technology. Please, next slide. Thank you. So although MGI is a very young company, only about 12 years in operation, we began when BGI acquired the US company Complete Genomics in 2013. Complete Genomics held the patents for DNB-seq technology, which is still the foundation of our entire portfolio of sequencers today. In 2017, as you can see, we launched our first sequencers, the G400 and the G50. Following that year in 2018, and continuing the trend of population projects, the T7 platform was launched, increasing sequencing capacity for our clients and users. The following year in 2019, we opened our production and R&D plant in Latvia, a milestone for the EU-based manufacturing, as it now supplies reagents and materials across the Europe continent. In 2022, when the pandemic struck, MGI developed strategies to facilitate fully automated sample processing, launching automation platforms such as the SP100, the SP960, and the sample transport platforms. Building on the knowledge generated in our R&D departments, in 2022, we officially launched the new versions of the G400 and the T7 in the EU, featuring improvements in chemistry and usability. Moreover, MGI has become a reference in the production of CE-IVD certified equipment and reagents for clinical applications, thanks to the highest certifications granted by international organizations, demonstrating the quality of the products we make available to our partners. In 2023, we launched the fastest platform in the market, the G99, as you can see. This sequencer is capable of sequencing PE50 in just 12 hours, designed for applications requiring low to mid capacity with up to and over 80 million reads per run. Most of our customers actually report more than 80 million reads. In 2024, our portable E25 sequencer was equally released, and we are currently presenting our latest platform, which is a DNB, we are currently promoting, which is the DNB-seq T1 Plus. I'm sure some of you must have heard about it already. This one integrates conversion, DNB generation, and flow cell loading, all in one equipment, and is equally CE-IVD certified, following the highest quality standards for both the instruments and the sequencing reagents. Next slide, please. Yeah, this slide is showing a little bit of what we do. So beyond the development of new sequencers, MGI is also a key player in the field of multiomics, incorporating into our portfolio not only DNA and RNA-based studies, but also proteomics, single-cell analysis, epigenetics, radiomics with ultrasound systems, and intelligent storage systems for samples. MGI's main areas of activity can be categorized into 3 groups. We have the seq-all, which focus on obtaining genomic data through our sequencing platforms. Then we have the prep or the GLI, which constitutes our automation branch that we used for strengthening sequencing through the automation of routine processes. Then we have the omics branch, which covers the multi-omics field, including single cell, spatial transcriptomics, ultrasound systems, and smart storage, smart sample storage, please. Sorry. Next slide. The next slide is simply showing the various sequencer, the various equipment across the three branches that we focus on. So we have a plethora of equipment that our customer can choose from. Next slide, please. Today we'll focus on the seq-all area. The technology used in MGI sequencers is based on DNB-seq technology, which was developed by the Complete Genomics and acquired by BGI in 2013. This technology relies on the generation of DNA nanoballs, as you can see. It's like a ball, but it's not actually a ball. So it relies on DNA nanoballs through rolling cycle replication or amplification, if you want. The double-stranded library is denatured and secularized. Then using the 529 enzyme, replication of only the template strand is achieved, reducing PCR duplicates and false positives and negatives as little as 3%. Is there still an echo?
No, the audio is fine.
Okay. So this significantly improves sequencing efficiency as fewer reads are wasted, directly lowering sequencing cost. In essence, the DNBs amplify the signal of the original template, reducing error propagation in downstream analysis, critical for somatic studies and liquid biopsy applications. Once the DNBs are generated, they are loaded onto our pattern flow cells that you can see here, where the DNBs attach to amine-coated spots through simple chemical reactions. External DNB preparation ensures cluster quality and avoid free adapters or overclustering that could disrupt sequencing. Let me add here that it is thanks to this technology, this unique technology, that makes MGI different from all other technologies, all other sequencing technologies. And actually, in fact, I will be bold to say that makes MGI the top or a key player or the top player in sequencing, because studies have been made that compared our sequencing technology to other sequencing technology, to our competitors. And most of the studies reveal that MGI sequencers produce better quality results. If you want, I could give you links for such papers. Please, next slide. It's an animation slide. As you can see, it shows the the RCR process. The advantage of this DNB technology are there's a reduction of PCR duplicates, like I already said, which allows for a higher effective sequencing rate. In other words, no cartridge reads are wasted. Next, we have reduction in index hopping, because performing DNBs outside the flow cell eliminates free adapters that will match other samples. This is very important in somatic and liquid biopsy applications. And then we also have the advantage of the fact that there is a great reduction in error rate, because the original template is always copied. Unlike in PCR, where the resulting daughter strands are copied, and then if there's an error that was incorporated downstream, the error is amplified in subsequent cycles. So we have here that if the 529 enzyme incorporates one error, that error is diluted within the total number of of copies because that error is not continued, is not copied, is not amplified further. And that's the big advantage of the DNB-seq technology. So thanks to the 1 DNB to 1 amine spot ratio on our flow cell, the potential for overclustering is equally eliminated because our flow cells are patent. So the quantification prior to loading the sequencer does not need to be as precise as with other sequencers. Next slide, please. So if you look at our portfolio, MGI offers 3 groups of platforms. I mean, our seq-all portfolio, we offer 3 groups of platforms. We have the fluorescence-based sequencers, which include the G99, the G50, the G400, the T7, the T1 Plus, the T7, and the world's largest sequencer, which is the T20.
Elvis, sorry, I have to interrupt you because the slides are not synced what you're describing. So maybe we need to go back.
So which slides? Sorry.
Okay, we see the T1 now.
No, skip this slide, please. I think it's a slide before. You went faster. Go behind. No, go 1 slide behind. Yes, this is where I was.
Okay.
Yeah. Okay. So then we have, so I said we have the first group, which is the G99. These are the fluorescence-based sequencers, the G99, G50, T7, T1 Plus, and all of that. Then we have the luminescence-based sequencer, which is the E25, the smaller one at the side there. Then we have the long-read sequencing, which is a nanopore sequencing technology, nanopore technology, which was recently developed, but not yet launched in all territories. So this broad portfolio reflects our philosophy. Every lab has different needs. That is why MGI offers sequencing solutions from small capacity instruments like E25 to benchtop platforms like the G99 and G400, and to high capacity systems like T1, T7, and T20. Additionally, our IVD certifications under new EU regulations strengthen our commitment to providing clinically validated solutions. Next slide. I think that will be my last slide. Next slide, please. Yeah, this one. Thank you. So essentially, we offer comprehensive training to all our customers and partners on the effective and efficient use of platforms vis-a-vis the applications or interests. So at the same time, we are leveraging our robust remote and on-site support tailored to your needs. So we equally offer support with your experimental designs. You know, if you have interest and you want to do, like, you know, if you're interested, whatever applications, not just reproductive, whatever applications, you know, we are well present, you know, very ready to assist you with designing, you know, experiments and the sequencing flow, workflow and all of that. Thank you. Next slide. I think the next slide is thank you. That will be all. So I look forward to getting questions from you. Thank you.
Okay, thank you, Elvis. Thank you, Renee, for the support. Again, if you have any question, feel free to drop them in the chat box. I can see that all questions have been answered so far. For some of them, we'll get back with some additional information. So there's no pending question. I see many guests are requesting for the presentation. So I guess we can collect them and send them through. If there is no other question or remarks from our speakers, then I think we can end the session here. Okay. Thank you everyone for participating in today's webinar, and we'll see you soon.
Thank you so
Thank
much.
you.
Thank
Thank
you.
you.
Have a good
Thank
day,
you.
everyone.
Bye-bye.